Hi guys, welcome to another video in biology. This video we're going to be highlighting the phospholipid bilayer. So the phospholipid bilayer is located on the outside of the cell. If this circle symbolizes my cell, then the phospholipid bilayer represents the border, the dark blue line that surrounds the cell. And that blue line creates a barrier for the environment inside the cell, which is different from the outside of the cell. Okay, so we're going to kind of zoom in a little bit on that blue line and look at what exactly it is about it that makes it so special and makes it uh, what about it makes it a good border for a cell. So on either side of the phospholipid bilayer is water. So that blue line that I showed you in the circle, that blue line is we're now zoomed in, and you're seeing the phospholipid bilayer right there. You have the blue dots, the blue circles with green lines. That represents that blue line in the circle. And of course, you see other stuff that's embedded in it that we're going to discuss because it's not a solid blue line. It's not solid matter. There's a lot of components. There's a lot of things at work in that blue line, in that border, in that phospholipid bilayer. So that's what we want to zoom in on. So right now I'm zooming in on a transport protein. These are proteins that are embedded, and there are several kinds of these proteins that are embedded inside of the phospholipid bilayer. Some of them go all the way through the phospholipid bilayer. Some of them go halfway through, and they all serve different functions. And we'll get to that at the end of the video as far as what these different functions are. But do understand that there are different types of proteins that are embedded inside of the phospholipid bilayer. And then we also have things that are embedded inside of the phospholipid bilayer, and that's cholesterol. Now, cholesterol is interesting because we've always thought and we've always been taught that cholesterol is a bad thing. Well, sure, too much cholesterol is a bad thing, but in general, cholesterol is something you need in your diet. Now, actually, I kind of take that back because your body actually produces the cholesterol that you need already. So, a few things about cholesterol. Um, Cholesterol helps to keep the phospholipid bilayer stable. It helps kind of reduce the effects of the different temperatures that we expose ourselves to. It helps to keep it more fluid, and it helps to keep it more rigid at the same time, depending on the temperature. Lastly, we're looking at the glycoprotein. Glycoprotein, this is a protein that is embedded on the transport protein itself, and that serves also a couple of other different functions in the cell, which we'll get to later. But the glyco part, just think glycolysis, glucose. Glyco is referring to the fact that there's a carbohydrate present in the structure of this protein itself. So here we are, we're looking at all the different components in the phospholipid bilayer. We have the outside of the cell, and then we have the inside of the cell. So let's dive in a little bit closer and look at the phospholipid itself. The phospholipid has two main components. Actually, it has three, but we're going to just highlight two or break down into two components just to make it a little bit more simple. The blue part is our hydrophilic head. Hydro meaning water, philic, like the city of Philadelphia, the city of brotherly love. This is hydro-loving. This is water-loving. This is the part of the phospholipid that's exposed to water. Okay, and why is it that it's exposed to water? We'll get to that in just a second. And then you have the green part that's called the hydrophobic. Phobic, phobia, a fear. So this has a fear of water, or this is the part that repels water. So let's dive into the hydrophilic part of this. So there's three components to the hydrophilic part: choline, phosphate, and glycerol. So it's not just like a round circle. We're getting in the nitty-gritty details of what these things are made out of. All right, so the choline. As I look at the choline molecule, you'll see here, oh, I'm sorry, and then of course you have the fatty acid chains at the bottom. Okay, so let's dissect each component here. The choline, you see the different elements that it's made out of. That's what the choline part is, and then the phosphate is a phosphate group. This is the part that gives us the negative charge. So remember, this is a polar head. This is the side of the phospholipid that is polar, which means one side of the molecule is negative and the other side is positive. And that's what makes it attractive to water, because if you remember, water is also polar, right? So the phosphate group is the part of the molecule that makes this this polar head, and it gives it a negative charge. And then you have glycerol, which is another hydrocarbon compound. And then attached to that at the bottom, you have your fatty acid chains. Now notice, these are all made of all carbons. Now, of course, I don't have the hydrogens shown here. And there's a couple of things that I want you to look at. First of all, look at the most obvious part of this, is that one of the fatty acid chains is bent. There's a kink to it. It's unsaturated. The unsaturated part of this is it's called the, it has the bent part to it. And it has the bent part to it because of the double bond that I have highlighted there. That double bond creates, it decreases the angle at which the top part is separated from the bottom. And then if you look at the left, you have the saturated aspect of the fatty acid chain, and of course that's saturated, meaning that it's got a full complement of hydrogen atoms attached to it. Now on the right side, the unsaturated, we had to take out a couple of hydrogens in order to accommodate that double bond, which created that band. Okay. So again, this is my hydrophilic head and the hydrophobic. Okay. So the hydrophobic part is the part that is, as you'll notice, as you notice the structure here, you'll notice that all the green fatty acid chains are facing inward, and then the polar heads are facing outward towards where the water's at. So that creates a border between the inside and the outside of the cell conveniently, and then of course I have the other molecules that we were discussing earlier. Now you notice the red arrows that I have. I'm pointing towards the our polar heads. These polar heads are constantly moving. They're exchanging positions laterally. If you could imagine like a lunch line, people changing every second, every nanosecond, people changing positions. And that's what happens with these polar heads, is that they're so fluid that they're constantly just moving positions along the lateral end next to each other. So, and then you have the nonpolar tails. These rarely switch positions. I read somewhere that maybe once every month or so you'll get the nonpolar fatty acid chains to switch spots, but for the most part it's a pretty rare occurrence. Unlike the polar heads that are constantly changing, our fatty acid chains don't really change that often. I want to finish up the video with highlighting some of the functions of the embedded proteins that are in the phospholipid bilayer. Now, as I mentioned before, there are many types of proteins that are embedded, and this is where we get the name for the fluid mosaic model. Think of mosaic being multicolored. There's multiple aspects to it. Well, it's the same thing with the surface of the cell. There are different types of proteins because the cell has many functions that it does in the body. So I'm going to highlight a couple of these functions. The first one is enzymatic activity. Enzymatic meaning enzymes. So there's enzyme activity here where, if you recall back from the previous chapter, enzymes can either make new compounds or they can break up compounds to make new ones. So, for example, here I may have a molecule that fits into that protein, which will then make a different molecule, which will then fit into the next protein, and then it'll make a brand new one that will be used by the cell in some way. So you'll notice that these substrates are constantly changing because of the proteins, the enzymes. And some of them are multi-step like this, and some are not. Some will just go into the enzyme and then it'll come out a new product. But sometimes that new product will need to go to a different protein that will then make the actual final product that will be used. Kind of like an assembly line, if you will. Sometimes the assembly line is long, sometimes it's short. Depends on what needs to be made. All right, so another job of the embedded proteins is cell-to-cell recognition. So I have here the phospholipid bilayer of one cell, of cell number 1. But if I have another cell that needs to find out, like for example, let's say you have an antibody that wants to that's going around looking for a virus. Well, it's going to go to each glycoprotein in the cell to see if it's a cell that belongs here, because you might have bacteria that are in your body that don't belong, and your immune system will identify that bacteria by the glycoproteins that are outside of its cell membrane. So remember that The glycoproteins serve as kind of like an ID card for the body to recognize that, okay, yes, you belong here, or no, you do not belong here, and then it goes about destroying that invader cell. Okay, and then you have intercellular joining. So you notice that in your body, your body is solid. So there's not, things don't just go through it. So you have all your cells are adjoined one to another, and you'll notice here on the proteins that I have shown here that they're in contact with each other, and they create what's called tight junctions. And these junctions are like locking mechanisms that keep the cell attached to its neighboring cell. Okay, so this is intercellular joining. And then finally, they sometimes can serve as attachments to the cytoskeleton. The cytoskeleton, if you remember, is the organelle in the cell that basically gives it its structure. It's its framing work, if you will. So it's like when you're building a house, the first part of building a house is the framework. Well, that's what the cytoskeleton is. It gives shape to the cell. It's like the skeleton, the cytoskeleton. And these proteins can serve as attachment points, as anchor points to that cytoskeleton. Okay, well that does it for now for the phospholipid bilayer. I hope that this video helped in your understanding of this concept. Good luck in your study.